Base pair abbreviated as BP. You will find BP for base pair in genetics books. Two complementary nucleotides that are paired in double-stranded DNA. Two complementary nucleotides are adenine complement is thymine and the guanine complement is thymine. Adenine pairs with thymine and guanine pairs with cytosine. A BP base pair is also used as a physical distance of length of a sequence of nucleotides. For example, 20 base pair or 20 BP is a chain of DNA composed of 20 nucleotides. Kilobase, kilo means 1000, 1000 base pairs of DNA or RNA. A mega base pair, 1 million, 10 to the power of 6 base pairs of BP. When you say base pair, adenine pair with thymine, cytosine with guanine in double-stranded DNA. Complementarity is the chemical affinity between specific nitrogenous bases. So adenine and guanine are adenine, guanine, cytosine, thymine, uracil are nitrogenous bases. So their affinity. Adenine, the affinity of adenine for thymine, the affinity of guanine for cytosine. This makes what the complementarity. So the chemical affinity between specific nitrogenous bases as a result of their hydrogen bonding properties. From where this affinity or the interest of one nucleotide or nitrogenous bases to the other is because of their bonding property, that is hydrogen bonding property based on their structure. This is a complementarity, adenine with thymine, cytosine with guanine, thymine with adenine, cytosine. Generally, AT and CG, simply, that we call complementarity. Then it will make the double-stranded DNA. Here, CG, AT, for example, GC, TA, AT, we call this complementarity. cDNA, C for complementary, so complementary DNA, a DNA copy of the messenger RNA, that is mRNA transcribed from a gene. The cDNA or complementary DNA is made from the mRNA using the enzyme reverse transcriptase. When there is transcription, DNA used to make RNA, that is mRNA. In this case, mRNA, the reverse process, mRNA is using as a template to make a DNA. Here is messenger RNA. Then from messenger RNA, finally we will get what? Complementary DNA. So messenger RNA will be reversed by reverse transcriptase enzyme. Then single-stranded DNA will be made. Then by DNA polymerase and complementarity base pairing process, there will be finally double-stranded DNA. Reverse transcriptase, this is an enzyme, an RNA-dependent DNA polymerase that synthesizes DNA from the RNA template, used by retroviruses like the human immunodeficiency virus or HIV to make proviral DNA from its RNA genome. DNA polymerases, enzymes that can synthesize new DNA strands using previously synthesized DNA or RNA as a template. So, what is the function of DNA polymerases? Making DNA molecule will be many. That is why it is called polymerase, DNA polymerase. By bringing nucleotide and making bonded with the other nucleotide and making the strand. This is a function of the DNA polymerase.